over to you, Abhishek.
Yes, thank you. So, good afternoon, everyone. My name is Abhishek. I am a final year PhD student, and my work is investigating embryo malformations, and I look at the coupling of calcium signaling and mechanics in embryogenesis. So, getting right into it, what motivates me to do what I do? So, the coupling between calcium signaling and mechanical forces is quite important in fertilization and embryogenesis. So, when you disrupt that calcium signaling, it can give you embryo malformation. So you see up top, that's a Spina Bifida case with the baby where it's a problem in the development of the central nervous system and the spinal cord hasn't formed or closed properly. So that's a Spina Bifida problem. Something far more serious is anencephaly. That's the case on the lower picture, and that pretty much results in death. And whereas like you have cases where like babies with less serious case of spina bifida, they survive, but anencephaly is fatal. So, of course, that makes calcium signaling very important. The mechanochemical coupling between calcium signaling and the mechanics is poorly understood, and few mechanochemical models exist at present. So, my PhD is to develop the models that couple the calcium dynamics and the mechanics. So, what is it that we are modeling? So, in short, the apical constriction of the anterior neural plate. Now, what do those terms mean? So, apical constriction is the contraction of the apical side of the cell that causes it to take on the wedge shape, as you will see in the picture above. So, you have the apical side, and say, for example, you have a cell with a fixed volume. So, if you have just one side of the cell shrinking, it'll take on a wedge shape. And because all these cells are connected to each other side to side, you have this sort of bending take place. So that's the process of apical constriction. What it does is, you see this row of cells up top, they make up a tissue plate called the neural plate. So once apical constriction begins happening, it folds this neural plate tissue and it brings sort of two ends of the tissue together. And then what happens is you have a sort of like tubular shape at the bottom and like a closed plate-like structure at the top. So the plate-like structure at the top, the blue one that you see below, that's the epidermis. And then the bottom one is the neural plate. So the neural plate is a key structure in chordate embryos. And this goes on to form the central nervous system. So the brain and the spinal cord, the neural tube goes on to form those. So the anterior plate that we are looking at, that goes on to form the brain, and the posterior neural plate goes on to form the spinal column. So the problem I'm looking at, as I mentioned before, like if the calcium signaling is disrupted for the anterior neural plate, you get cases like anencephaly. So here's a video from one of our collaborators. So it's a choreography of this cell autonomous calcium flashes, and this happens in conjunction with the tissue contraction during the neural tube closure process. So those flashes that you have, they used a gecko red dye, and that shows where calcium activity is happening. So whenever there's like an elevation of calcium, you have these red flashes. I'll just show that again. So wherever you have these red flashes, those are calcium flashes. And that happens, as you can see, wherever there's like elevated calcium activity, the tissue sort of like pulses over, that contracts. So this is another team whose work is very similar to the work that you saw before. So our collaborators from the University of Cyprus, they had done the previous work, and then came 2 years later, we had this work from a team in Japan. So their work had like experimental bits and mathematical modeling. So the experimental, you're seeing the phase towards the end of the neural tube closure, and you'll notice that the calcium flashes are more focused towards the sort of like the lips of this closing structure, and like you have the more frequent activity as you approach the end of this. So these are some of the key experimental observations, that in the regions where you have more folding, you have higher calcium activity, and also towards the later phases of the neural tube closure process, you have higher calcium activity. So it sort of like the calcium activity sort of speeds up the neural tube closure, and that has an effect on the calcium. So there's a two-way coupling that happens, and we'll look more at that later. So that sort of folding structure that you saw, it starts with this furrow formation in the neural plate. And as you form this furrow, then you go on to form the neural tube, as you see in the lower right picture over here. So a quick definition, a calcium flash or a transient is a temporary elevation in the level of cytosolic calcium concentration. So that's the calcium concentration of the cell level. So to sum up, the experiments show that the calcium fluctuations precede cell contractions during neural tube closure. There are two types of calcium transient patterns. One is a single cell transient, which lasts for a short time. And then you have this multicellular wave-like pattern. So it can exist in groups of, say, 2 to 5 cells and spread out over like the neighboring cells. So it can go over like tens of cells and propagate over a longer distance. Whereas a single cells just originate and die in the same cell. The calcium induces the activation and remodeling of these actin structures within the cell. So usually you may have heard of the term actin and myosin. So these are proteins that make up your muscles and lead to like muscle contraction and expansion. But in the cells, these actin structures, they sort of form like the cytoskeletal structure. So remodeling these would help you change the cell size permanently. So when the calcium activation happens, the mesh-like F-actin structures, they're remodeled, and this prevents the cell from recovering to its original state once the contractions happen. So the spatially and temporally random calcium transients on the single cell level have been noticed to have a greater impact on neural tube closure on the multicellular waves. So I'll focus on this kind of modeling. So just a quick look at the biology. So imagine the structure that you see on the right is a very rough diagram of a cell. So you have the cytosol, which is the cell body and the fluids within the sort of cell body. And if you were to model the level of calcium within the cell, you could have, say, a diffusion term, and then you could have like the different calcium flux, the influxes and outfluxes. So you have, for example, in the standard formulation of calcium flux modeling, you have these different terms called the J channels. So this is basically calcium flowing out from this internal calcium pool to the main body of the main body of the cell. So when we say a calcium flash has occurred, it can be due to two reasons. So for example, say you squeeze the cell and that causes an inflow of calcium from the outside. So that is the JSSCC term. So SSCC stands for stretch sensitive calcium channels, and they're exactly as like they do what they say they do. So if you stretch the cell, then there's a calcium influx and that elevates the calcium level. Or if there's like some sort of other chemical signal that the cell receives, then you have a calcium flow from the endoplasmic reticulum, that's this internal cell like calcium pool, to the main body of the cell. So that also causes the calcium elevation. And then, of course, the calcium, once your signals die down, the calcium flows back into either the pool or maybe it just leaks out of the cell. So if you're looking at a continuum modeling perspective, this is the equation you'd be looking at, but we look at a different kind of modeling, and we'll get into that later. So that's called agent-based modeling. So another thing in the biology that we need to look at is this ratchet-like mechanism. So you remember the mesh-like F-actin structures that I mentioned that were preventing the recovery of cell size. So those red spikes that you see on the graph are calcium flashes. And you'll notice that after calcium flash occurs, there's a sharp decrease in the cell area. And then there's a subsequent recovery of the cell area, but it never quite gets back to its previous pre-calcium flash state. So when the cell area and perimeter decrease below a certain threshold value, they cannot recover the original size. And this is what we'll call the ratchet-like mechanism. So the experimental results we want to capture with the model, the main ones, are calcium transients precede contraction. The oscillation frequency increases with time, and the tissue area decreases, and the oscillation amplitude increases with time. So we look at the vertex model, which is a cell-based on agent-based model. And it's an example of an off-lattice model. So you have on-lattice models and off-lattice models. So on-lattice models deal with a fixed grid size, and within those grids, you'd imagine those grids to be occupied by cells. And you can say that a cell is migrating or moving about if you say a cell stops existing in one grid and moves to the next. But because we want to focus on this change in cell shape, we look at an off-lattice model. With an on-lattice model, you can only you can focus on like cell shapes, but it's more coarse, whereas off-lattice models give you more freedom to visualize the cell shape and the tissue shape. So we modeled the neuroepithelial tissue that I mentioned earlier. We modeled the neural plate as an epithelial tissue, and we model the apical surface of that tissue because we want to capture the phenomenon of apical constriction. The cells are modeled as polygons, and the adjacent cells, as you can see, share nodes and edges. If you think from the graph sort of terminology, you can change the cell shapes by adding or removing nodes, or by lengthening or shortening the cell edges. So the model, it's based on the work, as if you remember from one of the videos by that team in Japan, so called the Suzuki model. It's an extension of the standard vertex model formulation because they've added equations for the ratchet-like mechanism. So we model the epithelial tissue, apical surface, as a 2D sheet with 271 cells. And we assume that the vertices move in a viscous medium, so the motion is overdamped. The energy minimization has three primary components. So the cell edge tension that's activated by calcium, that causes cell contraction. Then there's the cell perimeter elasticity that causes the cell dilation, the cell area elasticity that causes cell dilation again, and the ratchet-like mechanism that prevents the cell size recovery. And I'll explain the mechanism in greater detail as we go down the slides. So just at a quick look at the equations behind the vertex displacement. So we have AN and PN, which give you the area and perimeter of the cell. And AN naught and PN naught are the natural area and natural perimeter of the cells. So what does that mean? So there's a sort of like tug of war going on between the line tension and the elasticities. So the line tension at the cell edges wants to sort of contract the cell, whereas the elasticities want to restore area and perimeter to the natural area and perimeter values. But of course, it can't get all the way there because of the tension. So you settle at this sort of like tug of war, like steady state between the tension and the elasticities. So this gives you the force balance on every vertex. And as you evolve your simulation, you'll see the force on each of the vertices. They'll cause the vertices to move. So going in sequence, when a calcium flash occurs in a cell, that increases the line tension. So the calcium transient increases line tension by this constant amount, and this causes the cell edge length to decrease. So if you had this sort of like balance between the elasticities and line tension, a calcium flash would break that balance and cause the cell to suddenly shrink. So we determine this like quite randomly. So there's like a probability of occurrence of a calcium flash. So at every time step in the simulation, there's a probability that a calcium flash would occur, and the line tension would be elevated. And it's held elevated for as long as we think the calcium flash is lasting. So the line tension is held elevated for the calcium's activation time. Then the vertices are displaced to minimize the energy function that you have. So you have the three components: the area elastic potential energy, the perimeter elastic potential energy, and the line energies. So all three of them combined, they give you the total energy of the network, the total energy of the tissue. And then using that, you could find the force on each of the vertices, and that gives you the equations of motion for the vertices. So as I said before, the line tension tends to minimize the cell area and perimeter, and the area and perimeter tend to return to their natural values. So this is more of a visual aid to help you understand the ratchet mechanism. So remembering that graph that we saw with the calcium spike that causes the cell area to decrease sharply and then sort of recover. You have, in the first picture, you have the cell sort of settled between the sort of red line, which is the natural area and natural perimeter that it would like to be at. And the tension is sort of holding it away from that. So the black line gives you the cell's current area and perimeter. The yellow line gives you the sort of threshold. And if you shrink beyond that threshold, what happens is there's an irreversible change that takes place. The ratchet mechanism kicks in, and it basically corresponds to the F-actin structures remodeling the cell so that it can't recover to its original state once the calcium flash has died down. So there you have the equations of motion for the vertex once you expand all of that outwards. And it's you have the position vector for each of the vertices, and they have the X and Y coordinates. And you can see that the area of the cell, the perimeter of the cell, we model them as functions of the X and Y coordinates of the vertices. So we use a forward Euler scheme. This is keeping in line with the simulations done with the team in Japan. So we use the forward Euler scheme and it's quite sufficient to model the change in tissue shape. So how I went about doing this was I made the software package for off-lattice models in Python. So there exist a lot of agent-based modeling softwares. Most of them are on lattice. A few are off-lattice. And one of the notable ones is Chaste. Chaste have a lot of features, but it can be quite tricky to use. And I like to have a bit more control on the software package that I'm working with. And at the time when I started making this, I was ambitious enough to think that I'd develop a 3D vertex model. It's safe to say it's beyond the scope of my PhD now. It can be done, but it's beyond the scope of my PhD now. So we will can it there. But yeah, so how you go about doing the software development, the process I followed was I tested the code blocks in Jupyter Notebook, which is great for like just having spare bits of code lying about and just testing blocks of it and then moving it on to an actual IDE to make the full script that I want for the simulation. So Python has a large number of modules and packages, and you can just easily look up any sort of issues online and pretty much have the answers instantly. So one of the packages that I used was Network X, and that's basically for if you want to do any graph-based modeling. So the data structures I had were cells and vertices. So the vertex graph just had attributes of position for each of the vertices, and then you have the cell graph that had the attributes you get for each of the cells. So the area, the perimeter, the natural area, the natural perimeter, whether the calcium flash is whether the cell is in an activated state or a deactivated state. And there's a countdown timer. So when the cell is activated with calcium, you have a certain amount of cool down time before the cell gets deactivated again. So all of these states are stored within the sort of cell graph. And you can just store them very easily as attributes of that in those graphs. So the current features in the software package is that you have a tissue generator that generates a hexagonal lattice. You can either generate the tissues in this sort of like hexagonal, you can have these hexagons arranged in a hexagonal manner or this sort of rectangular manner. The numerical schemes that currently implements are forward Euler, backward Euler, predictor, corrector. For parameter sweeping, it has a feature to make most use of like multiple cores by parallel processing. And of course, you have data visualization because there's no point generating all the data if you don't know what it looks like. So in the future updates, once again, ambitious. Use Voronoi tessellations to generate more random tissue shapes because to more accurately reflect what the biology looks like. Then I also intend to put in like topological transitions, but for the current problem that I have, I don't need topological transitions, and also the 3D cell modeling that I mentioned earlier. So starting with the simulations, for the single cell case, we have the green being the calcium flash, and then the calcium flash brings about the shrinkage. So you'll see whenever the calcium flash is occurring, the cell shrinks, but once the flash goes away, the cell recovers. But it's not quite at its previous size. So over time, you have this irreversible change in cell size, and it just steadily goes on decreasing over time. So we get these pulse contractions as we had from the experiments of our collaborators from Cyprus. You'll see it in the top right graph. So they have the calcium flashes and the pulsed contractions. For 271 cells, you have, at the single cell level, you'll notice a sort of pulse contractions, but the tissue on the whole has a sort of monotonic decrease in area. So I just let that play out. See it get to the steady state. So it shrinks a lot quicker towards the beginning, then it sort of begins like plateauing out towards the end, and then it just reaches steady state. So we for it to reach steady state, we need to sort of artificially limit the ratchet-like mechanism to sort of hit a bottom value. Otherwise, you'll just shrink to zero, and then the simulation just gets very unstable. So the scheme converges for the forward Euler, as you've seen. So for the case where you have no calcium activity, you'll see that for the different time steps, the lines, they overlap. And there's like a slight deviation for the 0.8 case, but then once again, the scheme converges. So calcium, you'll notice if you compare both of the plots that calcium speeds up the contraction. So whereas earlier in the no calcium case, you were having this tissue reach steady state around like the 10,000 mark, whereas for the case where you sped up the tissue contraction with calcium, it happens somewhere around like 5,000. So what is our target? We want to select the parameters that will shrink the tissue to 2 to 8% of the original size in 40 to 60 minutes, which is what the time frame for the apical constriction during the final stages of the NTCR. So we go about doing this by performing a parameter sweep and quantify how the key mechanical parameters affect tissue shrinkage, and that gives us an idea of how the parameters depend on calcium. So the sweep was done for smaller tissues with 91 cells because there's a high computational cost for the higher cells. So now that it's currently in progress because the parallel processing was implemented very recently, and it's currently going on. So I'll have new results soon. So increasing the calcium flash frequency gives us that at low frequencies, if you raise the value of the calcium flash frequency, it sharply speeds up the constriction. But at the higher frequencies, say beyond 0.1, it pretty much plateaus out, and you basically don't get an effect on how long. It doesn't have much of an effect on how much time is taken to reach the steady state. And this matches the results that the Suzuki team had in Japan as well. Increasing the area elasticity, it slightly slows down constriction and increases the final tissue size. Increasing the perimeter elasticity greatly slows down the constriction and increases the final tissue size. Increasing the viscosity slows down constriction in a sort of linear fashion and has not much of a change on the final tissue size as you'd expect from viscosity, because that just slow the movement of the cell vertices and not actually have an effect on the final size. Increasing the baseline tension greatly speeds up the constriction and decreases the final size. And increasing the calcium dependent elevation in line tension. So that's expressed as like a percentage of the baseline tension. I must apologize because the formatting has been messed up in quite a few places. So do ask me if something is unclear, please. So increasing the calcium dependent elevation in line tension, that speeds up the constriction and decreases the final tissue size again. There's a similar effect to the baseline tension. So to sum up what all you just saw, so you'll see that the elasticities, if you increase the elasticities, that reduces the apical constriction. If you increase the viscosity, that reduces the amount of apical constriction that occurs. And if you increase the tensions, that increases the amount of apical constriction that will occur. So far, the vertex model captures the fact that the calcium spikes precede the tissue contraction and that the area decreases monotonically over time. So we still need to put in the frequency dependence over time. And currently, the calcium flash is modeled just as an on-off, active or deactive binary state. But we need to vary the amplitude over time and see if that has a significant effect on the time taken to shrink and the final tissue size. So the further work, as I said, would just be that. And also, because currently you have the tissues, you must have noticed, is just a set of cells that are sort of just in free space, but you don't generally get that in biology. You have free cells attached to some sort of epidermal layer. There's some boundary cells bounding it at least. So we'll be modeling the epidermal layer bounding the neural plate and see how that affects the tissue shrinkage again. And the ultimate aim is to develop a two-way model that has a two-way mechanochemical coupling between the calcium and the mechanics feeding back into the calcium, because currently you only have calcium affecting the mechanics, but no feedback from the mechanics to the calcium. And that would hopefully shed some more light on the embryo malformations. So thank you.
Yeah, that's great. Just have it on. I think question was that What would
Right.
calcium signaling everywhere?
Right. That's a good point, actually. During this part of the process, you don't have cell proliferation or cell death. So, and also, in fact, there's a reason. Usually, you'd have seen vertex models. You have the topological transition that takes care of some of the cell instabilities when like sizes get too small. We don't have that in this part of the process. So this process is like it's very limited in terms of what can happen in this part of the process. So you don't usually have cell birth, but I can tell you what happens in case of like what happens if cells get too small. We're using a 2D surface to approximate a 3D phenomena, right? So what happens if like a certain cell's apical surface area gets too small? The wedge-like cell is sort of like eliminated. Yeah, it just sort of shoots off in the opposite direction. Then you have this surface closing without it. It would be interesting to see what happens if you have like the constriction, but you also have like cell proliferation in that, how that would affect the rate.
Yes, you have this stuff. With the
right. With yes. Yes, sir. So going over the data, what I found was you have these sort of like changes in the sort of behavior of the tissue shrinkage because of the natural area and natural perimeter values. So usually what happens in the sort of like the first kink you get in the graph, that happens because say the natural area value has stopped changing. I can tell you what happens for like the second graph. So I know for a fact that over here, the natural area value stops changing because it's hit its sort of like floor. And then the natural perimeter value sort of hits its floor over here. And because they both hit their floors, the natural area and natural perimeter, that's why the tissue stops changing altogether. So in the earlier bits, because natural area and natural perimeter both hadn't hit their floors, the ratchet mechanism was active for like both the equations. That's why you have the sharp shrinkage. Whereas in like the sort of middle phase, you have the ratchet mechanism only active for the perimeter. That's why you have a more gentle shrinkage. And then because the ratchet mechanism is not active at all, it just plateaus out. It's something similar for the first graph as well. But I need to check because the calcium affects the rate of the shrinkage. So once you surpass like a certain speed of the tissue shrinkage, you get the sort of qualitative change in behavior of the plot. So you get either one or the other. So if I were to say increase PC slowly from 0 to 0.0001 or 0.001, as I go up there, that'd be one point where you have this qualitative flip. But then I need to look into like what value, what regime it is, where it's happening exactly.
This kind of seems like doing a lot, like a certain amount of cells in your sample. But I'm guessing you don't really have any transfer of things between the cells. Single cell, self-coupled, normalized way that need to become this.
That's actually, I do have an equation for the cell. So for like the single cell case, that gives you like a steady state equation. So based on like the elasticity and line tension values, you get the exact steady state size. So I'm trying to use that to gather some insights towards the parameter sweep. So because like if I show you the number of parameters I have, I wonder if I can show you the slides. That's just blanked out. How do I get back to my there. So those are all the parameters that I have that I'm working with. So of course, for like the multi-cell case, it's a bit difficult to figure out what's going on. Like I just hold all the parameters constant and just vary one, and then I get the plots as you've seen before. So for the single cell case, I have the steady state equation, and then I just study how those parameters are affecting the single cell. And then what I'm trying to do is gather some insights as to like what parameter regimes might affect the behavior for the multi-cell case. But I need the data from the simulations for the multi-cell case to draw any sort of like correlation between the two. So that's once again currently in progress. That's why the parallel processing bit is like very important, because like otherwise it just takes a bunch of time for 271 cells.
Blockchain, normal, a bunch of additional. Yeah, time dependent. Probably the time dependence, right?
Yes.
Yes. There's that Capgemini. That's that.